All right, so you're welcome to this presentation. We are going to talk about uh, biochemistry, and this is basically an introductory lecture. So, by the end of this presentation, you should be able to define biochemistry, you should be able to identify and describe the major classes of biomolecules, and then understand the role of micromolecules and their you need to understand the roles of micromolecules in biological systems. And then we'll look at one element of the periodic table called carbon. So you will understand the properties of carbon and um, what is the composition uh, of living organisms, and why carbon is very an important component of living organisms. So biochemistry is a branch of science that deals with chemical processes and substances occurring within living organisms. So living organisms are made up of substances, and these substances undergo uh, breakdown, or they can be synthesized. So with the biochemistry, we are looking at the nature, that is the structure of these substances, and how they function in the biological system or in our bodies. Okay, that is the interest of our presentation. So basically, look at how they look like, how these uh, molecules look like, how they work, and how they interact uh, with each other, and what are those factors that enable these reactions to take place. Looking at the enzymes, what enzymes are involved in these processes, what are the limiting factors for these reactions, and things like that. So that is basically the focus of biochemistry. So why do we really need to look at biochemistry? It's that we need to know that biochemistry um, acts as a bridge for our understanding of uh, pathological processes in the human body. That um, the nature, the quantities, the quantity, the structure, and the functions of these substances or these biomolecules can be altered. Then there is alteration in structure, in function of these biomolecules, then that means that a disease will happen. Okay, so if we understand the normal structure of these biomolecules and how they work, it gives us it provides us with a basis of understanding how diseases can be prevented and how diseases can be treated if they occur. Okay, so we also look at the other aspect, that actually drugs act or have influences on biomolecules. So that kind of interaction, if we understand the kind of interaction between the drugs that we give the patients and the biomolecules in the bodies in the body, it gives us hints on what medications we have to give, what dosages, and what basically are we really targeting. So pharmacology as a as a branch of health sciences has a biochemical element. Like we look at the interaction of drugs with the uh, with biomolecules, and what factors actually influence this kind of interaction. So that is basically point number two, is that we get to understand how drugs interact at a molecular level. And this interaction is very important in us uh, developing the right drugs and knowing um, if the drug has been administered, how does it interact, and what are the factors that actually influence its interaction, and what is the outcome of this interaction. Those are both all pharmacological aspects related to drugs, related to biochemistry. Okay, so biochemistry is a linkage between different health sciences. It links us to pharmacology, as we have seen. It links us to even nutrition. Okay, the food that we eat, the carbohydrates, the vitamins, the proteins, the lipids, all these have impact on our body. But understanding the way the body actually handles this food that we eat requires biochemistry. So biochemistry is very important in the different fields of nutrition, of nursing, and in of public health, okay, of medicine, of even dentistry, of pharmacy. Okay, so biochemistry is an important subject in life sciences. So the branches of biochemistry include what we call structural biochemistry, which is basically looking at the structure of these molecules that have are of biological importance to our bodies, and the micromolecules that basically we talk about are the proteins, carbohydrates, nucleic acids, and then lipids. We'll look at each of those categories in terms of their structure. We'll look at their function. We look at their breakdown. We shall also look at synthesis of some of their derivatives. Okay, so structural biochemistry is concerned with looking at the three-dimensional structure of these important biological micromolecules. Okay, so a number of techniques are used to be able to look at the structure of these molecules. You can use X-ray crystallography, or you can use the nuclear magnetic resonance, okay, or the electron microscope. These are some of the techniques that can be used to visualize the structure of these macromolecules. And then the other branch of biochemistry is what we call the metabolic biochemistry. With this, we want to look at the, the pathways or the chemical reactions involved in metabolism. So, for example, if we want to look at the processes involved in energy production, which enzymes are involved, which substrates are involved, and under what conditions do these reactions happen. The branch of biochemistry that deals with that is what we call metabolic biochemistry. So we are looking at metabolic reactions or metabolic pathways, which are basically chemical reactions taking place within the body that involve a number of substrates, a number of enzymes in different parts of the body, in different parts of the cells. And then molecular genetics. This one explores the relationship between genes and proteins and how they encode. Okay, the genes are the ones which determine the nature of proteins that are synthesized. So the study the study of the relationship between genes and proteins is what we call molecular genetics. So the most important aspect here is studying how protein synthesis is actually regulated. Okay, We also look at issues to do with the nucleic acid replication. In this, we look at basically the DNA replication, the RNA replication. Okay, so these are all processes that we shall look at. We shall look at the structure of the nucleic acids. We look at the bases, that the basic structure of the nucleic acid and the nucleotide, the bases, the pentose sugar, and the cytosine and the cytosine and the other bases that we shall look at. So the other one is clinical biochemistry. With this, we basically, for example, if you fall sick and then you go to see a physician, uh, they will request for, let us say, your blood. Okay, so they run tests to see the enzymes, some enzymes, the concentration of some enzymes, the markers, some markers in your blood, and other things like that. These are important in diagnosing diseases. So the field of biochemistry that is concerned with analysis of body fluids and tissues in order to make a diagnosis, is what we call clinical biochemistry. So a number of techniques are used: immunoassays, chromatography, spectrophotometry. These are examples of techniques used in clinical biochemistry. Then biochemical toxicology. So if you accidentally or intentionally take a toxin, or if there is an investigation related to whether someone was poisoned or not, then experts in biochemical toxicology have to be involved. These experts will actually look at the possibility that there are poisons, or someone has been poisoned, and how those poisons actually work within the body. What are the pathways, and what are the mechanisms that these poisons actually cause tissue damage or even cell death, okay, and their impact on the environment. So investigating toxins in relation to how they affect the body is a branch of biochemistry called um, biochemical toxicology. And then we move to the sixth, which is bioinformatics. Okay, this one is a combination of biology, computer science, and statistics. So we have three disciplines here: biology, computer science, and statistics, to analyze and interpret biological data. Okay, biological data such as DNA sequencing, looking at protein structures, then are looking at what we call systems biology. Okay, because these molecules that make a large part of our bodies contain information. Your DNA contains information. The proteins, the genes. This is all information. So this information, which we are basically calling biological data, needs to be collected and analyzed and interpreted such that it can be used for a number of reasons in science, in health. Okay, so the discipline that is concerned with looking with interpreting this biological data is what we call bioinformatics. So we have been talking about biomolecules for a number of times. Now, when we talk about biomolecules, we are basically referring to complex, large molecules that are formed as a result of combination of smaller molecules or units called monomers. So that means that biomolecules are large molecules, and these molecules are formed as a result of combination of monomers. So therefore, we can say that the biomolecules are, um, are polymers. When you have monomers combining, we form a large chain, which we call a polymer. Okay, so these biomolecules they consist of the carbohydrates, which are basically the energy sources in humans, and they include molecules such as glucose, sucrose, lactose, cellulose. These are all carbohydrates. The key thing is that they provide us with energy, but they are also important in certain structures of the cells. And then we move to the lipids, which also act as energy stores, but they also play an important role in regular in insulation and form part of the cell membranes. And then we look at the proteins, which are basically which carry out a number of reactions. I mean, a number of functions. They're involved in chemical reactions as enzymes. They're involved in communication as hormones. They are involved in supporting like the keratin, and then also on our skin, even our hair. These are all structure proteins. Okay, they are very important. So we also have issues like or aspects like antibodies. These are all proteins. So proteins carry out a number of functions in the body, and they are also biomolecules. And then we move to the last category, which are the nucleic acids. Okay, the nucleic acids are of some. Please, if you enter the classroom, make sure you're muted. Mute. When you enter the class, please mute. Okay, we don't want to have noise. All right. So, looking at the nucleic acids, these are of two types: the DNA and then the RNA. We will also look at the structure of the DNA and RNA, how they are actually replicated. Okay, how they are replicated. What happens if the DNA is damaged? What is the role of RNA? These are things that we'll look at. So basically, nucleic acids store and transmit genetic information. So earlier on we said that biomolecules are formed as a result of combination of monomers, and the process is actually called polymerization. Okay, so for example, when you have a carbohydrate, the carbohydrate can be as a result of combination of monomers such as glucose. So glucose combining to form a compact or a long chain carbohydrate. Okay, so therefore the carbohydrate is a polymer that is formed as a result of combination of monomers. So these biomolecules, the ones we have talked about, the lipids, the proteins, the carbohydrates, and the nucleic acids, all of them contain the following: one, carbon, hydrogen, oxygen, and then nitrogen. And of course, they can contain other elements, but basically these are the major ones that are found across all of them, whether it is nucleic acid, whether it is proteins, whether it is lipids, okay, whether it is carbohydrates. They all contain the four elements: carbon, hydrogen, oxygen, and nitrogen. Now, carbon is actually very, very important in this aspect. And we are going to look at why carbon is actually very unique. Carbon as an atom, or carbon atoms generally, form the backbone of all these organic molecules. The the biomolecules that we have talked about, the proteins, the lipids, the carbohydrates, we can call them organic compounds, okay, or organic molecules. So the carbon atoms form the backbone of these organic molecules. So carbon is very unique because it can form a number of bonds with with itself and also with other atoms. And we have four types of these bonds. Okay, we have covalent bonds, we have polar and non-polar bonds, and then hydrogen bonds, and then ionic bonds. These are the ones we are going to need to focus on. Okay, so one, looking at the carbon. Carbon has a symbol C. So if you see C in a biochemistry book, it means carbon. It has an atomic number of 6 and an atomic mass of 12. Okay, atomic number is number of protons. Okay, and then atomic mass is number of protons and neutrons in an atom. Okay, and then we move to the other aspect of carbon is that it occurs naturally in different forms, including graphite, diamond, and amorphous carbon, and it also occurs in organic compounds that we have seen. The proteins, proteins, carbohydrates, and then the lipids, and nucleic acids. You will always find carbon there. Okay, it has a valency of 4. Okay, this is that it has 4 electrons in its outermost shell, and this therefore means that it can form up to 3 covalent bonds. Okay, I mean up to 4 covalent bonds. Okay, so these covalent bonds can be single bonds or double bonds or even triple bonds. Okay, so when we have single bonds, you see a dash. This dash that you see here, C and a C, and then in between there is a dash. That is a bond. It signifies a bond. So you can have one carbon atom, for example, in methane, covalently bonded to 4 hydrogen atoms. That is why you have CH4, that is methane. And then ethane, you have 2 carbon atoms connected to 6 hydrogen atoms. Okay, but the connection between carbon to carbon is a single bond, and then the connections between carbon and hydrogen are also single bonds. Okay, then we have double bonds. This is where you have the 2 dashes here, C and C. In between these 2 dashes, that signifies a double bond. Okay, so ethane is an example of a compound that has double bonds. Formaldehyde also has double bonds. And then we have triple bonds. Okay, acetylene is an example of a compound which has triple bond. So these bonds can be polar or non-polar depending on the electronegativity between carbon and the atom with which it bonds with. Okay, so remember, in covalent bonds there is sharing of electrons. Covalent bonds are formed as a result of sharing electrons. So when carbon shares its electrons with an equally electronegative atom, then that bond is non-polar because it does not result into a resultant positive or negative charge, and therefore it is a non-polar. If carbon shares electrons with another atom that have different electronegativities, then you will have a polar covalent bond. So for example, the bond between carbon and oxygen, okay, this this bond is polar, okay, because oxygen is more electronegative than carbon. And then the other type of bonds, what we call hydrogen bonds. Carbon itself does not form hydrogen bonds directly, but in organic molecules where you have a compact structure, you will see uh, hydrogen bonds being formed as a result of electronegativity difference between the molecules or the atoms. For example, uh, um, electronegative atoms like oxygen and nitrogen. Okay, this will result into hydrogen bonds being formed. Hydrogen bonds are very, very important to understand, especially in biomolecules and their structures. Issues to do with the tertiary and secondary structure of proteins, or even looking at the structure of DNA, you will look at the hydrogen bonds in the DNA and also in the proteins. And then we have the ionic bond, which is the other type of bonds. These ones are formed when you have atoms that have um, different electronegativities. So highly electronegative elements like halogens, like fluorine and chlorine, combining with metallic ions. Those bonds, for example, the bonds in sodium chloride are ionic because you have strongly or highly electronegative elements combining with metal ions. So hydrogen bonds are the ones that we have just discussed. Electronegativity, we have just been talking about it over and over again, but it is a simple concept. It's that you have atoms that don't have the same uh, the same charge, okay, and therefore, because of the difference in charge, electrons will be attracted towards uh, an atom of high electronegativity. Okay, so if you have a covalent bond between, let us say, carbon and oxygen, that means that electrons have been shared, but these electrons will be drawn closer to oxygen because oxygen is more electronegative. So that is the concept of electronegativity. Okay, it helps us in predicting the polarity of chemical bonds. So that means that if you're going to have a bond between compounds which have similar um, uh, electronegativities, then you're not going to have these electrons drawn more towards one of the atoms. And therefore, electronegativity influences a number of physical and chemical processes that biochemical molecules undergo. So that has been our lecture. If you have any questions, you can now ask.